Hi folks, my name's Dr. Paul Rose, and this presentation covers key pointers about observational methods that can be used for behavioural research. It's by no means exhaustive, but it covers key tools, key techniques, and key terminology associated with the collection of observational data on the human and on the non-human animal. At first glance, recording behavioural data and observing what individuals are doing might appear challenging. Individuals can live in large groups, they can look similar, they can coexist with different species within a complex and dynamic environment. But understanding your research question, selecting the appropriate population, and ensuring you follow a consistent method will allow you to collect valid and reliable data. The techniques we use for observational recording do not just apply to the non-human animal. They can also be used to study human behaviour too. Here we've got two populations, one that's human and one that's the non-human animal. I hope you can see that individuals of both species look different. They have different characteristics that we can use to tell them apart. We can follow individual behaviour patterns, or we can follow the behaviour of the group collectively. Depending on what your question is, and depending on what you wish to find out, that will dictate how and when you record behaviour, and who you select to record behaviour on. Are you interested in what the individual is doing specifically, or do you care about what the group is doing overall? Either way, so long as you know what method works best to answer your particular hypothesis, you will be able to use the same types of recording and sampling techniques for observing the behaviour of the human animal, as well as recording and observing the behaviour of the non-human animal. Let's start off with some key considerations. Are you interested in the individual or the group? I've already mentioned this when looking at what individuals can be doing compared to what the group might be doing overall. Once you've decided on your sample population, you then need to make sure you understand the behaviours that the species you're focusing on will perform. You must produce an ethogram, which is a list of behaviours with definitions that species is likely to perform. The original ethogram that you use for data collection might be much larger than an ethogram that lists only the behaviours that you actually observed that goes into your final report or paper. You then need to think about categorising the types of behaviour that the animal will perform. And we call these behaviours states or events. And both the human and the non-human animal perform state and event behaviours. And the last, but probably the most important consideration, is putting together your sampling protocols with your recording methods. Sampling is who you watch and what you measure, and recording is how you go about watching and when you take measurements. The type of sampling protocol and the type of recording protocol depend very much on the behaviours, the states or the events that you wish to measure. Those state and event behaviours are going to be very specific to the species of animal, human or non-human, that is the focus of your research project. If you can identify individuals and you can follow that particular individual and record exactly who it is, that is called your focal subject. Therefore, you are doing a focal follow of a particular individual animal. In the case of this swallowtail butterfly, individual patterns and markings allow the observer to recognise individuals and follow their movements. Perhaps we are interested in how long specific butterflies spend at different flowers. Is that consistent across the species overall, or is there individual variation in how those individuals forage? Identifiable characteristic markings that we can note down that allow us to follow particular individuals means that focal follows work best in species where each individual has identifiable specific characteristics. Of course, in the field, it might be very difficult to determine who is who. Animals, individuals within that species or population, might look incredibly similar. Here is a flock of grey-lag geese. It's a mixture of adults with the white triangles on the base of their tail compared to goslings, which are smaller and fluffier. Once you get your eye in, individual identification can become easier. But if it's harder or impossible, we might have to do a group scan. That's different from a focal follow because you're not following and timing the behaviours of a particular individual. You're scanning across a group and counting the number of individuals that you see performing specific behaviours. Always use a group scan if you cannot identify and therefore follow particular individuals. It's important to get the distinction between the two basic categories of behaviour. This cow is chewing the cud. This is a state behaviour because it takes the animal a long time to perform, and it is therefore the state that the individual is in. State behaviours, long duration behaviours, can be timed in minutes or seconds, and therefore they can be put into a time activity budget to work out how much time, and therefore energy, and therefore motivation, the animal, human or non-human, is spending on a particular behaviour. Overlaid on top of these state behaviours are what we call event behaviours. Short, very quick, momentary actions that we count and display as a frequency or as a rate of occurrence. Going back to the flock of grazing geese. Grazing is the state behaviour that the geese are engaged in. But watch every now and then an individual sticks its head up and scans the immediate environment before going back to grazing. This alert behaviour is counted. It occurs too quickly to time, so we can time the grazing because the state lasts for longer. But we count the number of alert heads up we see whilst we're timing the grazing. Here's an excellent example of an event behaviour. Specific, discrete vocalizations that can be counted once we hear this bird performing them. To be clear on the difference between your state and event behaviours, categorize each action that you see the animal performing in an ethogram. An ethogram is a list of the behaviours that species performs. It provides a definition of what the behaviour looks like so others, if they were observing that species, would be able to work out what precise behaviour is being performed. In the case of this play behaviour in these chimpanzees, we wouldn't describe play, which is the category of behaviour, as playing. We would explain what play looks like. We would also explain that play is a state behaviour because it takes up a long period of the animal's time, like the resting and grooming behaviours that we can also see in this chimpanzee colony. Always construct your ethogram before you start any behavioural observations. That way you know exactly what behaviours to look out for, which behaviours are likely to be performed, and how you can identify them. Once you have your ethogram, and once you know what sample population you are going to follow, and whether or not the behaviours you are observing are states or events, you can think about your sampling and recording procedures. Sampling rules are who you watch and what you measure, and recording rules are how you watch and when you measure. And there are different types of sampling and recording rules that you can use for observational studies. Ad lib sampling is used to define your ethogram. you record everything that you see the animal doing, and then you go back and give descriptions and definitions. Focal sampling is when you follow the behaviours of a particular individual in a group. Scan sampling is where you look across the group and you count the number of individuals you see performing particular behaviours. Behavioural sampling is when you simply record the behaviour that you are most interested in. This is commonly used for very rare or unusual behaviours. And event sampling is the type of approach where you are interested in just recording these short, instant behaviours that you can't time. Recording techniques can be continuous. You get a continuous amount of time that the animal spends on that particular behaviour. They can be instantaneous when you record, for example, on the minute, every minute for an hour, or they can be 1-0, where you say yes or no, that behaviour has occurred within a preset defined time period. We can put these different sampling and recording techniques together. Perhaps we're interested in the amount of time these viewers watch the giraffes at this zoo. We could record the behaviour of particular individuals in the crowd. That could be a focal follow. So that is a focal sample, and perhaps we could record the time spent viewing the giraffes continuously. So we've got a focal continuous recording method. Maybe we're interested in how the giraffe overall change their behaviour according to the number of visitors. We could scan across the group of giraffes to record how often are the giraffes looking at the visitors. That would be an instantaneous scan sample of the group. Let's put all of that into a worked example. Perhaps your research question is individual differences in the time fish spend foraging. You found a suitable population of fish and you've realised you can individually identify them because they are differently coloured. Think about visibility. Can you always see what the animal is doing? Remember, out of sight is not a behaviour. Here is a handy checklist that you can follow when planning an observational study. Have you identified your research question and have you used that to formulate an aim and a hypothesis that you're going to test, which is the whole point of you collecting data? Have you identified what particular species you're going to use to test your hypothesis? Which species works best for the research question that interests you? What is your species likely to do and when is it likely to do it? Base your observations, your data collection period, around the activity pattern of your species. Be sure to have constructed an ethogram with clear definitions and descriptions of the behaviours that you're likely to see. And can you follow recognisable individuals? Can you do focal follows? Or will you have to scan across the entire group? That will dictate who and what you will sample, as well as how and when you will record data. And finally, think about how much time you will need to spend observing your population. Do you require a lot of data across a large number of individuals? Or can you answer your question with a smaller amount of data on a smaller number of individuals? Will you be doing replicates? Will you repeat observations on the same individuals? All of this needs to be planned and considered before you actually start your observations. Going back to the goldfish, what's our aim? To investigate the foraging behaviour of these fish. What's our hypothesis? It could be that differently coloured fish spend different amounts of time foraging. The more brightly coloured you are, the less time you might spend foraging because the more conspicuous you are to predators. What's our state or event behaviours? Well, foraging is a state behaviour. It takes up a large proportion of the animal's time. How will we define foraging? When we see the fish searching for food within the weeds and plants in the pond or from the water's surface. What other state behaviours could the fish perform? They could be swimming, they could be stationary, motionless in the water column, they could be interacting with the conspecifics, the other fish around them. All of these are states, so we can record behaviour as a moment of time and put that into a time budget. We know what we're sampling because we've worked out individual differences based on the fish's colour pattern. Some fish are multicoloured, some fish are white and red. So our sampling, who we watch, is a focal sample. What we're measuring is the foraging behaviour of the focal individuals. And our recording methods, because we're going to collect the amount of time, could be a continuous focal sample, an exact recording of the number of minutes or seconds spent on foraging compared to those non-foraging behaviours, or an instantaneous focal sample where we break up the observation period into specific time points and we record the individual's behaviour on each specific time point. This presentation has explained the difference between state and event behaviours. It's explained the differences needed for data collection on an individual compared to a group of animals, and it's provided information on how to construct an ethogram and define clear explanations of the behaviours being observed. Finally, it's covered the differences between sampling and recording techniques so we know exactly who and what is going to be sampled, as well as how and when behaviour are going to be recorded. It's really important to remember that states and events must be recorded differently because they're presented differently in the results sections of any papers or reports that generated from the research. Thank you very much.